Good morning, good afternoon, or good evening to everybody who has joined today, wherever you are in the world. Thank you so much for being here. First of all, I just want to take a moment to thank CyberSight and Orbis International for the kind invitation to be part of this live webinar. I've joined many of their other webinars, and you can see it's a very stellar lineup of speakers, experts in their field. So I'm just so incredibly humbled and honored to be part of this lineup. I am going to talk a little bit today about a really exciting area in the world of retina and honestly in the world of ophthalmology, because I think this is going to be spreading across our entire field and has the potential to help an incredible number of patients. So I hope you're excited. My name is Christina Weng. I am on faculty at the Baylor College of Medicine in Houston, Texas, in the United States. I'm a professor of ophthalmology there. I also serve as the fellowship program director for the Surgical Retina Fellowship Training Program here. And I'm going to talk a little bit today about the clinical and surgical considerations of gene therapy in retina patients. I sort of dabble in a lot of different areas, but thanks to my chairman, Timothy Stout, who's done a lot of incredible work in this area, when I started my career about a decade ago here in Houston, I started getting involved with gene therapy. And fortunately, the timing was just right because it was the beginning of a lot of important candidates coming through the pipeline. And I even saw one of those drugs see approval by the regulatory authorities here in the United States. So we'll talk about all of that today for about 45 minutes. I'm going to save about 15 minutes towards the end for any questions. So please feel free to type in any questions that you have into the Q&A function. These are my disclosures. Of note, I do serve as a consultant to several companies that do have gene therapy candidates, and I've also served as a principal and a sub-investigator for some gene therapy trials. Let me start here by this article that I clipped back in 2020. This comes from the FDA. The FDA, as many of you know, is the Food and Drug Administration. It's the regulatory authority here in the United States. It's the organization that essentially approves our drugs for patient use. In 2020, this release was given by the FDA that just really highlighted what a burgeoning space this is. At that point, 4 years ago, there were 900 investigational new drug applications sitting on the desk of the FDA. So just a lot of interest in this area. In fact, that year, the FDA actually had to hire 50 new personnel to join their team just to be able to review all of these applications. And actually, now that we're 4 years down the road, I'm certain that that number is even greater. So again, lots of interest and lots of research that's going on in this area. As you probably know, if you pay attention first at the middle column, there are many different types of gene therapy that exist, and we'll go through these in a short bit of time. But the type of gene therapy that any patient needs really depends on the type of genetic disease that you are trying to treat, which is in the first column. So let's look at the first row together, for example. Autosomal recessive. These are also known as loss of function inherited retinal diseases, where a patient has a mutation that they're not able to produce a protein that our body usually needs to function. That's an autosomal recessive loss of function inherited retinal disease. And the gene therapy type to treat that is gene augmentation, sometimes referred to as gene replacement or gene supplementation. This is the most common type of genetic disease, these loss of function mutations. And so gene augmentation was really sort of the earliest foray into gene therapy. And one example of this type of gene therapy is our one and only currently approved gene therapy in the United States, which is voretigene neparvovec, which is used to treat a biallelic RPE65 deficiency. Let's move to the second row now. What about autosomal dominant, or also known as gain of function inherited retinal diseases? This is where a patient has a mutation where there's overproduction of a certain type of protein. Well, to treat that, these are notoriously a challenging class of diseases to treat, and we haven't had as great luck treating this class of diseases because not only do you need to inactivate or suppress that gene that's overexpressing, but a lot of times, as you all know, we need some level of that protein. You can't just suppress it completely. And so a lot of times that has to be done concurrently with a low level of gene augmentation as well after completely shutting it down. It's a little trickier. Fortunately, we've made really great strides in this area for technologies that might be able to help us treat these gain of function inherited retinal diseases. in the form of small interfering RNAs and also antisense oligonucleotides. Another really exciting area that can be very helpful in this category of genetic diseases is probably something you've heard about a lot in the recent news, which is gene editing. We'll talk a little bit about that later in the deck, but this is one example of this is the CRISPR-Cas9 technology that some of you might have heard about, and that's being looked at for a variety of diseases, including LCA10. And finally, in the bottom row, we have multifactorial diseases, right? Diseases where you can't necessarily attribute the disease to a single locus. For example, macular degeneration. We know that there's a root in genetics there, but we don't know, there's not one single gene that can be pinpointed for macular degeneration. It's sort of a combination, and there's a large variety. What about that type of genetic disease? Well, we are able to now leverage some progress in the gene therapy world in the sense of biofactory approaches. And so if you think about this, for wet macular degeneration, how do we treat those patients? We treat those patients with anti-VEGF injections, right? We give these injections sometimes as frequently as every month. It's a lot for those patients. What if we could give them a gene therapy that would allow their own eye to produce its own supply of anti-VEGF therapy? How neat would that be? And that's exactly what's happening. In fact, we have several programs that are in pivotal stages. For example, the ABB-VRGX314, ADVM022, 4D150, and there's more than that as well. So really exciting area. The last type I want to touch on that is also excellent for honestly all of the different types that you see listed here is a gene agnostic type of gene therapy called optogenetics. I'll tell you a little bit more about that, but this allows sort of the cells that are remaining in the retina to potentially take over the function for the photoreceptors, which are often damaged in inherited retinal diseases. And one of those examples that is coming along in the trials is MCO10. So as you can see, bottom line, a lot of really exciting areas that are happening right now for all the different genetic disease types. How does gene therapy work? Let's take a 10,000-foot view at this schematic. So essentially, and this is a little bit oversimplifying, but you create a transgene that codes for the protein that you desire. And that transgene is packaged into a vector. The vector is going to carry that into the host cell. And these vectors can either be viral, which are the classic types we use, adenovirus, adeno-associated viruses, lentiviruses, or they can be non-viral as well, as I mentioned earlier briefly, antisense oligonucleotides, DNA compacted nanoparticles. These are all ways of getting that transgene into the eye. But anyway, whatever you package, you put into the vector is then taken with an endosome into the cell of the host, and then it breaks down. That vesicle releases the vector injects that new transgene into the nucleus of the host cell, and that allows the host cell to begin producing whatever protein is dictated or encoded by that transgene. And that could be either a native protein like RPE65, or it can be a non-native protein that our bodies don't typically produce, for example, an anti-VEGF fragment. We've become really sophisticated in these technologies such that the transgenes you can also play around with and modify. And you can actually, modify the promoter, the enhancer, the inverted terminal repeats, all of those things. We've really become more advanced in engineering the vectors. All of these different types of modifications allow for different behaviors and characteristics of the gene therapy. And it really opens the door to the applications that we're able to turn to these days compared to, say, 20 years ago. As you can see here in this non-comprehensive list, ocular gene therapy today targets many different diseases. Traditionally, when we talked about gene therapy, we thought about sort of those orphan and really rare inherited retinal diseases. And those are still on this list. I'm thankful to say we've made great strides in achromatopsia, choroideremia, for example, Leber congenital amaurosis, Stargardt, X-linked retinoschisis. Those are still on this list, and those are still a wonderful application of gene therapy. But I think what's interesting is that you'll also see on this list now a lot of our most common diseases. For example, diabetic macular edema and diabetic retinopathy, as well as wet and dry macular degeneration. This is by no means a comprehensive list, but just a snapshot that I found published at the end of 2023. That is actually, these lists change by the day because there's always trials that didn't work out and new trials that are being added. But just a small glimpse of all the exciting clinical trials that are happening right now in this space. You can see a lot of these are in earlier stages. In fact, this doesn't even include the preclinical trials, of which there's even more. They include a lot of phase 1 and phase 2 studies, but you'll also notice a lot of later phase studies in phase 3, some pivotal studies already that are happening. So super exciting. And it just means that we're going to hear a lot of buzz about this in the forthcoming years. How do you get gene therapy into the eye? Well, let me first start off by saying that the eye is incredibly fortunate because it's relatively immune privileged. It's a great space, right? Our eyes are sort of this compartmentalized area of the body. And so it allows different types of gene therapies to really do quite well. So we're fortunate in that we have that advantage over potentially different parts of the bodies where gene therapy is also being looked at. How do you get the gene therapy into the eye? There's really three main routes of gene therapy administration. Look at the schematic to the right of this slide. Initially, back in the day, we used to think that we would need to put the gene therapy as close to the target cells as possible. So for example, for LHON, Leber hereditary optic neuropathy, we thought that an intravitreal injection would probably be best because the cells we're really trying to target in that disease are the ganglion cells. Alternatively, on the flip side, for a disease like Leber congenital amaurosis, where the disease cells are primarily at the RPE, we thought, hey, maybe we should put in a subretinal injection for that condition because those, disease cells are closer to that space. But because of the advances that I just showed you on the last slide, where we're really able to modify our vectors better than ever before, we're able to play with the transgenes, this rule of thumb really no longer holds. And in fact, all three routes of gene therapy administration, suprachoroidal, subretinal, and intravitreal, are all being looked at, and I think all have validity, and they all deserve a good look to see which one of these is the optimal approach. And we may find that the optimal approach depends on the disease that we are trying to treat. Most of you are familiar with intravitreal injections, where we put the substance into the vitreous cavity. Suprachoroidal is the newest of the three different routes that are being looked at. This is given by a very short needle near the pars plana into the space that falls between the sclera and the choroid. So it's superficial, but it's been shown in animal and human study to penetrate quite deeply across all of the ocular tissues, even the posterior wall of the eye. And then finally, subretinal is the most well-studied of these three. And subretinal actually has two different subtypes. There's a transvitreal approach to subretinal gene therapy, and there's a suprachoroidal approach to subretinal gene therapy. The top left image that you see there is the transvitreal approach to subretinal gene therapy, where you actually go through the vitreous cavity, oftentimes performing a concurrent vitrectomy, and you inject it into the subretinal space. Contrast that to what you see at the bottom. That's an example of a suprachoroidal approach to subretinal gene therapy, where you actually tunnel a catheter through the suprachoroidal space. And then when you hit the targeted area, there's a little needle that emerges and enters into the subretinal space. And of course, one of the benefits of that approach is that you can avoid disturbing the vitreous, which can be really advantageous, especially in the younger patient populations. Like I said, all three are being studied actively as we speak, and all three of them each has their advantages and disadvantages. So if you look at the first row, intravitreal injections, we know those are generally very safe to give. They're very convenient to give because you can do it right there in the clinic. The drawbacks of intravitreal forms of gene therapy are they tend to be more immunogenic. They tend to incite a greater inflammatory reaction when we give them. It's something we need to look out for, and that's actually happened in some of the studies we've seen already, which we'll touch on. Additionally, the transduction efficiency, especially of the more common recombinant adeno-associated viral vectors that we use, AAV2 and AAV8, for example, seems to be maybe a little bit less. It might be limited by the internal limiting membrane and difficult to penetrate the deeper cell layers. Although, again, that's not a fixed statement because of the advances that we've made in terms of vector technology as well as the transgene engineering itself. The next two rows are talking about the subretinal approaches, both the transvitreal as well as the suprachoroidal routes. So the advantages of both of these are that they are less immunogenic. In fact, the subretinal space, I would argue, is the most immune privileged of all of these, and it tends not to be as inflammatory as some of the other routes that we're looking at. Additionally, if you go through the suprachoroidal approach, the subretinal gene therapy approach, you avoid the need for a concurrent vitrectomy, as I mentioned on the last slide. The drawback, of course, is the opposite of what I just said, which is that if you go transvitreally, it does require a surgery for the patient and it requires a concurrent vitrectomy. If you look at the suprachoroidal subretinal route, one of the disadvantages of that form is that it does require greater technical demands to sort of tunnel through the suprachoroidal space and then sort of penetrate this very thin membrane into the subretinal space is definitely not easy. And carries, of course, some unique risks to that. Lastly, in the final row, we're going to talk about suprachoroidal approaches to delivery as well. One of the advantages of suprachoroidal delivery is that it can also be given in the clinic and is fairly well tolerated by the patient. It may offer better transduction efficiency compared to the intravitreal route, although still to be seen. That's being actively studied. The drawbacks of suprachoroidal is just that we haven't had as much study as we have in the other areas. And so the immunogenic response is not quite as well understood. And additionally, injecting it into that space precisely has proven to be somewhat challenging in certain populations. And they're working on sort of different approaches to that and individualizing the needle length. So in the next section, I thought we would talk about 5 gene therapy candidates because there's no way I can go through that entire list. You can see there was just an incredible number there. But I thought I would pick out 5 that are either in late phases of clinical trials or that you're probably hearing a lot of buzz about right now in the news. So all of these you may have heard of or you will be hearing about very soon. I'm going to breeze through these very quickly, just kind of highlighting the top characteristics and features to know, and then we'll talk about each of these 5 in a little bit more detail. The first one is ABBV-RGX-314. This is an AAV8-based subretinal therapy that leverages that biofactory approach that we talked about earlier and is being looked at for wet macular degeneration in its subretinal form. So this gene therapy encodes for a non-native protein. It encodes for an anti-VEGF antibody fragment that's similar to ranibizumab, which we've used now for 20 years. We are also looking at this gene therapy to be given in the suprachoroidal delivery approach. We're looking at that both for wet macular degeneration and diabetic retinopathy. The next one on this list is ADVM-022. This is a modified AAV2 vector-based intravitreal therapy that also leverages the biofactory approach, encoding for a molecule similar to aflibercept, and is also being studied for wet macular degeneration. Of note, it was also being looked at for diabetic macular edema, but unfortunately, there were some adverse events that were noted, and that trial was halted back in 2021. So it's only moving forward for wet macular degeneration at this time. There are 2 that I've lumped together on that third row, AAV-RPGR and AGTC-501. And these are both similar therapies in the sense that they're both subretinal gene therapies that are being used to treat RPGR-associated X-linked retinitis pigmentosa. We'll talk about those in more detail shortly. 2 more to finish off this list. The first is MCO10. This is an AAV2-based intravitreal gene therapy that is gene agnostic. And I talked about this briefly on that one slide, right? So this introduces transgenes that produce proteins that are light sensitive called opsins that essentially use the cells that are remaining in the retina to act as photoreceptors. You can think of it that way in an oversimplistic description. And this is being studied in retinitis pigmentosa and Stargardt disease. The final one to round out this list of 5 is EDIT-101. This is a CRISPR-Cas9 technology. It's a subretinal treatment that is currently being looked at for LCA10. Let's talk about these in a little bit more detail now. Starting with the first, AABV-RGX-314. It's kind of a mouthful. Again, this is an AAV8 vector-based single-dose subretinal gene therapy producing an antibody fragment similar to ranibizumab. In the phase 1/2A studies, there were some really encouraging efficacy signals. They looked at 42 patients with wet macular degeneration and divided them up into 5 different cohorts as a dose escalating design, meaning lower cohorts got lower doses, higher cohorts got higher doses of this gene therapy. These were not treatment naive eyes. These 42 patients were high demand patients who had required anywhere on average between 9 and 10 injections in the year prior to entering the study. So they required a lot of treatment. And so they gave them one-time gene therapy with AAB RGX-314 and watched how they did. It was very encouraging to see that the visual and anatomic responses remained very stable. So they were already stabilized sort of entering the study because they had gotten so many injections. They remained very stable. But what was most impressive is the reduction in injection burden was almost 80% in cohort 5 at 2 years. It was well tolerated. There were some pigmentary changes observed in the retina, which we've seen in other subretinal gene therapies as well. We're still actively learning about that, but otherwise patients did quite well. And this has moved on to the pivotal phases. There's 2 ongoing studies called Atmosphere and Ascent that are looking at subretinal AAB RGX-314 for wet macular degeneration. And this is, as I mentioned, also being studied in a suprachoroidal form for wet AMD and diabetic retinopathy. Let me show you one of the highlight slides from that phase 1/2A study that I think is so impressive. So across the bottom on the x-axis, these are the different cohorts, the different doses of gene therapy. And as you go towards cohort 5 to the right of this slide, the gene therapy doses are becoming higher. And you can see in the gray bars, that was the number of injections per year that on average the patients were getting in each of these cohorts. To the right of those gray bars are the number of injections they were getting after the gene therapy. So for example, in cohort 5, you can see that at the 2-year time point, there was a 78.1 reduction in the annualized injection burden for these patients, a really meaningful difference. The next gene therapy I want to discuss in more detail is ADVM-022. So this is an intravitreal gene therapy, as I mentioned. And it uses an adeno-associated virus 2 serotype, but it's a modified AAV2. So it's very good at penetrating deeper layers of the retina, as we kind of alluded to earlier. It encodes for a molecule similar to aflibercept and has been studied in a phase 1 study called OPTIC that was recently just published. This also had really encouraging findings, just like the one I just showed you. And they took 4 cohorts, 2 high dose, 2 low dose, with wet macular degeneration, also a really high need population. They were treated also many, many times on average per year before entering into this study. They found similar findings. There was a stable to improved visual and anatomic response at 2 years. But again, the most impressive takeaway was the reduction in treatment burden by about 80% in the low dose cohorts, and even greater than that in the higher dose cohorts. And in fact, more than half of the low dose patients remained injection free at 2 years. So imagine you were requiring 9 to 10 injections, on average of anti-VEGF, and then you get the gene therapy and suddenly half of them don't need any more injections for 2 years. And that might be going on longer because the follow-up is still ongoing right now. Pretty unbelievable. There were some patients affected by inflammation in this study, in fact, quite a few. This tended to be low grade and responded well to topical steroids, and there were no cases of severe inflammation like vasculitis, retinitis, and choroiditis. But it is something to be aware of. And as I mentioned to you, these intravitreal gene therapies tend to be more inflammatory. Certainly that was seen in the DME study below called Infinity. And in fact, there were such serious cases of inflammation and hypotony in that study that the trial was actually halted. So it is something we're looking at. And while ADVM-022 has progressed to the phase 2 LUNA study, they've taken some precautions. In fact, they took just the lower dose from the OPTIC study to continue on. So it's a 2E11 dose that they're moving forward with. And they actually incorporated an even lower dose in the phase 2 LUNA study to try to lessen the inflammatory effects that were being observed. So they're also looking at a 6E10 gene therapy dose in that study. So we'll learn more. The second thing that they're doing are that they are incorporating some prophylactic steroid regimens that are being tested in LUNA that we're going to learn a lot about. Here's one of the slides from the phase 1 OPTIC study that's very impressive. At the top is the high dose cohort. The bottom is the low dose cohort. That vertical line in the middle where the syringe is the time point where the gene therapy was given. And so each of those dots represents an injection of anti-VEGF. You can see to the left of that vertical line, these patients were requiring a lot of injections in the year before they entered this study. To the right of that vertical line, after they received the gene therapy, you can see a dramatic reduction in the number of anti-VEGF injections that were required by these patients. So pretty impressive once again. I'm going to move and talk about this pair of gene therapies for X-linked retinitis pigmentosa, as I mentioned earlier. The first one is AAV-RPGR, also known as botaretigene sparoparvovec, which is an AAV2.5-based subretinal gene therapy that's used to treat XLRP that is associated with a mutation in the RPGR gene. This was studied in a phase 1/2 study, and at 6 months, they found some nice efficacy signals. They saw a mean retinal sensitivity that increased significantly in the treated versus untreated eyes. They also had a visual function test where patients had to navigate through a mobility maze, and they found that there was an improved time to completion of that maze that they were able to walk through in the treated versus untreated eyes. So well tolerated overall. The phase 3 Lumios trial is underway, and in fact, we are likely to hear a readout by the end of the year. The AGTC-501 is another similar gene therapy. It's also subretinal. It's also used to treat the same disease, RPGR-associated X-linked retinitis pigmentosa. In the phase 1/2 trial at 1 year, all patients treated centrally showed a positive microperimetry response, and this was also very well tolerated. The first patient was just enrolled for this AGTC-501 phase 2/3 trial called VISTA at the top of 2024, and so enrollment is actively underway. I'm going to move now to MCO10. As I mentioned, this is an AAV2-based intravitreal gene therapy that is mutation agnostic, meaning that no matter what type of mutation you have, you potentially could have benefits from this type of disease of gene therapy. And I really am interested and intrigued by this class of diseases because for a very long time, we've had monogenetic treatments, right? Where we basically find a disease, we find where the mutation is, and we see how we can address that mutation. That is great. And we've had, obviously, our first FDA-approved therapy is a monogenetic type of treatment. However, you can imagine how that's very limiting in the sense of resources and time and progress, because you can only look at one mutation at a time in those types of approaches. And yet we know that there are thousands and thousands of different mutations that are out there causing ophthalmologic diseases. And so looking at these gene agnostic types of therapies, I think, is a very, very important and could be very promising in this space. As I said, this intravitreal gene therapy is injected and allows the patient to produce opsins. Opsins are light-sensitive proteins that are usually found in the photoreceptors that sensitize and convert light into electrochemical signals that are then transmitted to the bipolar cells, eventually to the ganglion cells, and eventually it's what we see as vision. In these diseased eyes, these photoreceptors are no longer functioning or no longer functioning well. And so what MCO10 attempts to do is introduce a transgene that produces an opsin that then allows the eye to be sensitized at the bipolar cell level. So these patients, while their photoreceptors are not working, their bipolar cells may be working very well. And they essentially allow these bipolar cells to then function and detect light. That's the goal of this type of therapy, which has fast track and orphan drug designations from the FDA. It's currently being studied in a pivotal trial called RESTORE, which is a phase 2b/3 program looking at the treatment of retinitis pigmentosa. This is a 100-week study that is already had some preliminary readouts showing some very encouraging findings. So the primary endpoint was met, and that was a functional mobility test where patients had to navigate at different light levels through a certain maze. So that was met, but really impressively was one of the secondary endpoints that was pre-specified, looking at best corrected visual acuity, where they also found improvement in the patients that were treated with MCO10. Actually, both doses, they found improvements there at week 52 compared to sham. MCO10 is also being studied in a phase 2 trial for Stargardt disease. So it's really interesting because retinitis pigmentosa is the most common inherited retinal disease. Stargardt is the most common macular inherited retinal disease. And so these two are really encompassing a lot of patients around the world potentially. The phase 2 Starlight trial is looking at various outcomes as well. And so far, they've also seen some really encouraging findings at week 24, where patients on average had a BCVA improvement of 5.5 letters. And in fact, with a magnifier, that visual acuity improvement was 15 letters. There was also improvement on the visual field perimetry test in terms of mean sensitivity gain. And these results have been promising enough that a phase 3 trial is now being planned. The fifth and final one that I'm going to talk about is Edit 101, which is a CRISPR-Cas9 subretinal treatment. It's a gene editing system that you can think of as sort of molecular scissors. CRISPR-Cas9 is truly like science fiction turned true, but it's actually going. Some of you might have heard some recent buzz about a trial called Brilliance that was just published last month in the New England Journal of Medicine. We'll talk about that shortly. But essentially what this type of genome editing system does is it is able to find a sequence of mutated genes, cut it out, and replace it with a functional sequence. So cool. And like I said, this is being actively studied in several different areas, but one that I want to highlight for you is the phase 1/2 Brilliance trial, which included 14 patients, 2 children in that group, for a disease called Leber congenital amaurosis type 10, which is associated with a mutation in the CEP290 gene. They looked at various efficacy measures, including best-corrected visual acuity, FST or full field stimulus testing. They looked at patients' performance through a maze, a visual function navigation maze, as well as vision-related quality of life assessments. They found that 11 of those 14 patients experienced improvements in one or more of these visual and quality of life measures. In fact, many of these patients had improvements in 2, 3, or even 4 of these measures, which is really exciting. 4 out of the 14 patients had clinically meaningful best-corrected visual acuity improvements as well. So a lot of promise in this area. Still early, still warranting study, but it's been really encouraging to see that so far it's been well tolerated with no dose-limiting toxicities in this small cohort. One of them that is not in this list of 5 because it's already FDA approved and our only FDA approved gene therapy, I just want to spend a moment on because this is the one I have the most experience with. Viretigene neparvovec is trademarked as Luxturna. It was approved in the United States in December of 2017, becoming our first FDA-approved ophthalmic gene therapy. It's currently being administered in 17 sites of excellence in the United States. Baylor is one of them. We were one of the first actually to start giving this, which is why I have more experience in this area. And as I mentioned, this is a AAV2-based subretinal gene therapy consisting of 1.5 E11 vectored genomes contained in a volume of 0.3 cc's. And it's indicated and approved for patients with a biallelic RPE65 mutation-associated retinal dystrophy, which clinically manifests as Leber congenital amaurosis type 2 and certain variants of retinitis pigmentosa. The patient also has to be determined to have viable retinal cells to be able to receive this. One of the interesting facts about this drug that I want to touch on because it has implications from a social standpoint when we're talking about accessibility and care eventually as these gene therapy products become more widely available over the next few decades is the cost. It's an incredibly expensive drug. It's the most expensive drug that we have in the ophthalmology space right now. It's almost $500,000 US at this point. So it's incredibly expensive. And I hope that future gene therapies that are going to be forthcoming will have lower price tags additionally, but that will also find creative ways to ensure that as many patients who need these types of treatments are able to get them. The outcomes of Retina neparvovec, I just want to touch on because it's something that I get asked a lot is, well, you've given a lot of these patients, gene therapy. How are they doing? It's a great question to ask. We're so used to talking about outcomes and improvement in terms of Snellen letters on an eye chart, right? We bring the patient in, we have them read the eye chart, and that's what we consider outcomes for patients. And that is true for a lot of our diseases like macular degeneration, diabetic eye disease, etc. It's a little bit different when it comes to inherited retinal diseases because a lot of these patients may have low enough levels of vision that you may not be able to even assess their outcomes objectively very well on a letter chart. Additionally, there is more to vision, we know that, than just being able to read letters. It's also lots of areas of visual function that are actively being studied right now, but that we don't typically measure in our clinics on a day-to-day basis. And that was very true of the Retina Parvovec trials. So one thing that was super interesting and sort of landmark in their phase 3 registration study is that the primary outcome was not visual acuity. That was not how this drug, this gene therapy, became approved. It was actually based on patients' performance on something called a multiluminance mobility test. This is essentially a human maze. You set up an obstacle for the patient, you let them walk through it, and you see what their time is and what their successful rate of completing the maze is. And you test that at different light levels, since these patients with RPE65 deficiency have a very difficult time seeing even in slightly dim light, not to mention in darkness. They have a very difficult time navigating. And so that was the primary outcome in phase 3. And that was met, which is what led to the approval of Voretigene and Parvovec. So my point being is that we need to look at visual function more holistically, especially in this class of diseases. And in fact, that type of finding has persisted. So those patients in the phase 3 program, a lot of them rolled into the post-approval phase 4 study called PASS. And in the interim analysis of PASS, once again, we saw that patients treated in the real world actually, it wasn't an extension of phase 3. This is a real world study, patients who were treated post-marketing. There was no significant change in the visual acuity of patients, similar to what we saw in phase 3, but there was a mean FST decrease, meaning that patients were able to detect light more sensitively and able to essentially improve in that aspect of visual function. Additionally, myself and some colleagues, after this drug was approved, we sort of pooled our cases and published this a couple of years ago in Ophthalmology Retina in a multi-center case series of 77 eyes in 41 patients. We looked at various aspects, various factors that could impact mean visual acuity change at 1 year, including baseline visual acuity, whether we detach the fovea during surgery, the patient's age, et cetera. We looked at all of these things, and none of them seem to have a statistically significant association with the patient's visual acuity change at 1 year. However, what we did find is that those high gainers, those who gain 2 lines or more of vision, 16 out of the 17 eyes belong to pediatric patients. And I think one of the really important lessons that we're actively studying is that it makes a lot of sense that with these types of inherited retinal diseases that are progressive and degenerative, it's probably better to get in as early as possible and to treat these patients while they're younger, an active area of study. In addition to age, who else should be getting these gene therapies, especially when we have limited resources, right? We want those patients who are going to have the greatest likelihood of gaining visual function and having some of these benefits. We don't know. That's an unknown area that's actively being looked at. And in fact, there are many working groups of very bright people in this space that are developing consensus recommendations for who should be eligible for gene therapies. So it's an area that we really do need more minds to be put together. I just want to show you really quickly this multiluminance mobility test so you have an idea. This is really neat to see, but essentially you set up all these cushions in a very dark room, and these rooms have to be totally blacked out, meaning that all the windows are covered, even underneath the door it needs to be covered so that it's pitch black. And then we have different ways of adjusting the light to different levels, as you see in the scale below. And these patients are asked to navigate through this human maze. That's how a multiluminance mobility test works. There's many different versions of this that have been validated and are being used now in different trials, which is why I wanted to show it to you. This one was the one used by Spark Therapeutics for the Voretigene study, but there are others now that are being built into some of the other gene therapy trials. I want to show you, though, some of the challenges that we still face in this area. So let me show you 2 patients who I treated with Voretigene and Parvovec. They both have very similar looking OCTs, and yet the 6-year-old boy on the left had a very significant visual acuity improvement, as you can see down below, whereas the patient to the right had a much more modest improvement, didn't change as much. And yet again, I couldn't really see any differences preoperatively. This is really where we need help identifying which candidates are going to gain the most from these types of interventions. However, I also want to use this example to point out that vision goes beyond just the visual acuity reading. And in fact, we look at functional outcomes. And in children, especially, these functional outcomes might be things that you don't even think about. For example, the patient on the left had a very hard time going to sleep at nighttime because when it would get dark, he would start crying. He wouldn't want to go to sleep. His parents were really frustrated. And he also then wouldn't be able to wake up for school the next day. So he wasn't doing very well academically. After his gene therapy, they really saw this night and day transformation of this patient. He was able to function at nighttime, do his homework, and because of his good night's sleep, he was able to go to school the next day and started doing a lot better in that setting. The boy on the right, even though he didn't have a significant improvement in visual acuity, he was, for the first time in his life, able to go trick-or-treating, and his parents were just absolutely thrilled that he was able to do that, which was about a month after his gene therapy treatment. And those of you who aren't familiar, trick-or-treating is happens for a holiday called Halloween, which is celebrated in America, where kids go out at nighttime to collect candy from different houses in the neighborhood. And unfortunately, he was never able to participate because he wasn't able to see at nighttime. So he could never go and join his friends. And for the first time after his treatment, he was able to do that. Here are just some surgical considerations when you're thinking about performing a subretinal gene therapy. I'm not going to run through this in the interest of time, but I'll let you read them, and these slides will be available for you. There are certain preoperative things that you may want to do. For example, most definitely, you want to make sure you have genetic confirmation before you treat any of these patients. There are several different programs that allow for gene therapy testing. Two of them that I really like because they are no cost are Invitae and Prevention Genetics. So I've left the websites down below. You can order these kits and test your patients for a variety of different gene therapies. And I think that's increasingly important as we have more and more trials that are ongoing and potentially different treatments down the line. I think it's more important now than ever before for patients to understand what their mutation might be. Patient counseling is always important. As I mentioned, prophylactic corticosteroid treatment is a reality for a lot of these different trials that are being looked at. Ancillary testing to look at things like ERG, FAF, fundus autofluorescence, FST testing. These are types of tests that we don't maybe typically do on an everyday basis, but are very important for this group of patients. On the day of operation, I do prefer general anesthesia for these patients just to make sure that they don't move, especially for gene therapy treatment, and especially since a lot of these are younger children. And then post-operatively, you'll want to taper corticosteroids if you gave them corticosteroids. Oftentimes, we just use our standard post-operative topical drops. And for certain subretinal gene therapies like voretigene, I do like to position these patients supine for 24 hours just to let that gravity work and hold that gene therapy in the macula as much as possible. The instruments that you need and the supplies are actually not that different from what we typically use in the operating room. And I know that some of you had that question, but all you really need for subretinal gene therapy is a 41 gauge cannula so that you can enter into the subretinal space. And you can see the magnified view to the right. I like to bevel these cannula so that they enter the subretinal space a little more easily. You need some tubing to connect that cannula to the gene therapy, which is contained in a 1 cc syringe. I'm going to show you some videos now so you can actually see me performing this. This is a patient with RPE65 deficiency. You can see me starting off with the core vitrectomy. And then for these patients, because we're going to place this gene therapy into the subretinal space, we do need to elevate the hyaloid. So I use triamcinolone to stain the hyaloid, and then my cutter to elevate it, as you can see me doing here. If you don't elevate it, it makes the bleb, the subretinal bleb, very hard to raise. And that is why we do this. When you inject the gene therapy, you can either self-inject it with a foot pedal, if you connect it up to the correct apparatus, or you can have someone manually assist and inject that syringe while you're holding it. So here we go. I'm touching down along the supratemporal arcade. You see a little bit of blanching, and now you see that subretinal bleb being raised. It's 0.3 cc's. It's actually not that much, but this bleb looks humongous under the microscope. And I was so amazed when I gave this for the first time to see how large of a volume that was. You don't need to laser the hole that you've made. That actually will self-seal usually by the end of the case. So there's no reason to do anything there. And we deliberately do try to elevate the foveal center in this type of gene therapy, although it varies. Different types of gene therapy, sometimes you don't necessarily need to give it in the macula and raise the fovea. And I always like to suture in these cases to prevent egress of the gene therapy product to extraocular spaces. Here's another video, again in slow motion, just so you can see the blanching of the retina that I'm looking for. When I see that blanching, that's when I have my assistant start injecting, and that propelling of the injection actually opens up that subretinal space. Here's another video showing the actual injection speed, just so you can get an idea. It takes about 60 seconds to inject the entire 0.3 cc's into the subretinal space. And as I'm crossing the foveal center, I'll actually slow down the injection velocity just a little bit, just to make sure that we don't create an iatrogenic full thickness macular hole, which has been reported in subretinal injections. Additionally, after I finish giving the injection, I actually will keep inside the bleb. I'll stay there for a few seconds before withdrawing, just to avoid reflex and let that gene therapy settle. You want to minimize how much comes out into the vitreous space. Now, some people will consider pre-injecting with a bleb of BSS or even air. I haven't done that. I just like to really inject and make sure it's just the drug product entering that space, because we don't know what really happens with drug concentration or localization of that gene therapy, but some people have done that, so know that it is done. And once again, staying there for a few seconds before I withdraw, and that entry site made with the 41 gauge cannula will self-seal. Here's another case of me injecting an older patient. You can see by more of the pigmentary changes that they're an older patient, 30-some-year-old patient with RPE65 deficiency. And I'm using the help of a cool technology called intraoperative OCT that can tell you exactly where you are to confirm that you are indeed in the subretinal space. And the reason that's important is because in these older patients, it can be really easy to penetrate into the sub-RPE space, which you don't want to be in for these subretinal gene therapy injections. That's one really important application of intraoperative OCT. I just wanted to show you that. What are future directions as I wrap up here? So as I mentioned, intraoperative OCT I think is incredibly helpful for confirming the space that you're in when you're treating these patients. But I think another really cool application of intraoperative OCT is going to be with volumetric measurements of our subretinal blebs. So for example, when we inject into that space, you don't really know how much is coming out into the vitreous cavity. And in fact, some of my colleagues at Duke did a test in an animal model asking the surgeon to estimate how much of their gene therapy ended up in the subretinal bleb. And the bottom line is we found out that the surgeons were very bad at estimating how much was actually in that subretinal bleb. And so as we become more sophisticated and we begin refining our dosing, I think that knowing exactly how much is being delivered into the subretinal space is going to play a really key role in our treatments in the future, and intraoperative OCT can help us do that. Another really exciting area that some of you might have heard about is robotics, right? So this sounds like science fiction, but again, is another area that is actually being studied now. And in fact, just last year, there was a first in-human application of using robotics to deliver subretinal therapy. It wasn't gene therapy. It was TPA, tissue plasminogen activator, but it was put into the subretinal space. As many of you know and can imagine, entering the potential subretinal space is challenging, and you have to really be very careful in that delicate area. So if we have something that like robotics that can assist us in being more precise in injecting that subretinal gene therapy, it really could be a game changer in this area. And so I'm very excited about learning more about that as study continues. There are many questions that remain. As I said, this is a bustling area with a lot of study, and it amazes me that as far as we've come, there's still a lot of questions that we need answers to. Things like what the optimal delivery approach is. I told you, is it intravitreal, subretinal, suprachoroidal? We actually don't know yet, and that's all actively being studied. We talked about finding a good surgical candidate. How come some people improve more than others? We need to learn more about helping us identify those who are going to be the best candidates to be treated. We also need to know what the ideal age to treat is. We know we probably should be treating when patients are younger, but how young is that? Should we be treating newborns with these inherited retinal diseases? We don't know. What is the best surgical technique? Does hyaloid actually need to be elevated? And some people argue that it doesn't. We don't have an answer to that yet. The location and the volume of the blebs is something that is also being looked at. I talked to you briefly earlier that some of these subretinal treatments, including Voretigene Parvovec, as you can see below, that was published in a case series in 2022, are associated with pigmentary changes. And this happened not right away, but down the line after the patient was treated with Voretigene Parvovec, you can see all of those pigmentary changes along the arcades. Most of these seem to be clinically insignificant, meaning that the patient doesn't have any untoward visual impact. However, some of them are. And so, are these clinically meaningful? Why do they happen? We don't quite know yet. And it's an area of actively being investigated. One of the questions I've always had is, what happens as you have constitutively active transgene end product? I mean, have you ever thought about that? You put in a gene therapy, for example, let's take one that's a biofactory approach. Now the patient is continuously producing anti-VEGF, theoretically, indefinitely. You can't control how much, you can't shut that off at this point, right? Are there consequences to that? What does that mean? And what would happen if we needed to shut it off? Those are all very important questions to answer. Theoretically, because retinal cells are non-replicating, the durability of treatment should be forever. It should be indefinite. But is that really true? We don't have a long enough runway of study yet because these are such novel new treatments, but that is a question to be asked. And if it's not forever that they last, is retreatment possible? And finally, as I mentioned, the accessibility question. As more and more gene therapies become available in the upcoming years, how are we going to ensure that all the patients who need these types of treatments are able to access them? Very important. So in summary, retinal gene therapy has the potential to transform the treatment of both rare and common diseases. We only have one FDA-approved ophthalmic gene therapy at this time in the form of Voretigene Parvovec, but there's so many promising candidates in the pipeline, which is really exciting. And we're going to learn more from the slew of ongoing trials that I showed you, as well as real-world experience. We will gain additional insights regarding these therapies. I'm going to take a look at some of the questions that have come through now. So please feel free and continue to send in your questions if you have any in our final 10 minutes. Let me take a look here. I'm going to start with a safety question because I think this is such an important question to ask, especially as you venture off into these novel treatments where you're just not sure, and we don't have a long enough runway to really fully grasp safety over a period of decades, for example. And so one person asked, has there ever been a documented case of unanticipated dangerous genetic modifications from ocular gene therapies? And I think that's a really excellent question. As you probably know and are alluding to, there have been some off-target effects in animal models as well as in some other types of gene therapies outside of the eye. However, to my understanding and knowledge, there has not been any dangerous genetic modifications from ocular gene therapies that I'm aware of at this point. And I think there's a couple things to say about that. So first of all, the most common viral vector that you see being used is an adeno-associated viral vector. And one of the benefits of an AAV vector is that it doesn't actually integrate into the host DNA. It's episomal. And so that minimizes the chances of mutagenesis happening. And that's unlike, for example, a lentiviral vector. A lentivirus vector actually integrates into the host DNA, and that has a greater potential for mutagenesis. So that's a really important question. And additionally, we are going to learn more. So that's one of the potential concerns of technologies like CRISPR-Cas9, where you're looking at gene editing. And that's why they're also looking at other ways now of gene editing, something called prime editing, for example, that may have minimal chances of, or no chances of mutations being introduced into the host genome. So really excellent question. Thanks so much for it. Let me take a look here. Let me see. So I'm not quite understanding this question. It says, does age matter to apply laser on the blade port in subretinal injection? So I'm guessing that the question is whether age plays a role in whether or not laser treatment needs to be done. for the retinotomy site. And as I mentioned, it does not. And I've treated patients as young as 4 and as old as 39 with voretigene neparvovec. And in fact, the retina is very elastic to some extent and is able to seal these holes up very, very nicely. In fact, by the end of the case, the surgical case, which usually takes around 30 to 45 minutes, I can't even find the retinotomy site most of the time. So the 41 gauge cannula is very, very tiny. And no, I've never had to laser that retinotomy opening. So thanks for that question. How long before gene therapy will be clinically available? Thank you very much for that question. As I mentioned, in the United States and in certain parts of Europe, voretigene neparvovec, and also I believe in Japan, voretigene neparvovec is already commercially available. So we actually already have gene therapy clinically available for RPE65 deficiency retinal associated dystrophy patients. However, you're absolutely correct that there are many other types of gene therapies being studied that are still in early phases or in phase 2 or phase 3 studies where we don't have results yet. And so for those types of treatments, it still could be many, many years, if ever, that they come to market and become commercially available. I have heard that one of the farthest along is ABBV RGX-314. Their prediction is 2025 or 2026, where we're going to have top line data and potential approval. However, that is a speculation at this time because their full phase 3 pivotal study readouts have not been done yet. And so we don't yet have confirmation of their efficacy and safety, and it hasn't yet been submitted. So a good answer to that question is it will still be a number of years, I think, before we have a number of gene therapies that are commercially available for ophthalmic patients. However, a lot of these patients that we're treating are still very young, and it just takes that breakthrough. So it's very likely in their lifetime there will be opportunities to treat even the most common IRDs like retinitis pigmentosa. So that's what I tell my patients. Let me take a look here. I'm going to take one of the questions from Fabian Rios in Spanish. ¿En qué grupos étnicos es más efectivo el tratamiento génico? So I think my Spanish is a little rusty, Fabian, but I think you're asking that which types of ethnic groups is genetic treatment most effective? That's an excellent question because we know from other diseases, even our most common injections that we give in retina, anti-VEGF, we know that they potentially can have different profiles and effectiveness in different ethnic groups. And it's so important to understand that when we're talking about therapies like gene therapy, that they can have different effects and profiles in different races, in different genders, in different ages. Unfortunately, we don't have a great answer to that question at this point because the phase 3 studies for voretigene were quite small. So we really need larger scale studies, and we need to make more conscious efforts, I think, of recruiting diverse populations in these types of trials to better understand that type of question. But I'm so glad that you brought it up. Let me take a look here. One question from King Yip about subretinal injection models. There's a manual and an auto injection. Which one is better and what's the reason for the difference? That's a great question. Thank you so much, King, for that. So as I mentioned, when you're giving the subretinal injections in the operating room, you can either have a manual assistant injecting the syringe of gene therapy for you, or you can connect it to your foot pedal and actually use your foot pedal to inject the gene therapy on your own. There's definite pros and cons to both. So let me walk you through those. I've mostly done the manual injection, although I have also done an auto self-inject. The manual, what's nice about that is that it's very simple. You don't need any additional equipment. You just hand the syringe to your assistant. And as long as they inject slowly and are very well and familiar with the injection of subretinal gene therapy, which you don't want to suddenly inject forcefully, it often goes very, very smoothly and it's very effective and efficient. The drawback is that you need an assistant, and we don't always have that, right? And you need not just any assistant, you need a trained assistant who's really given gene therapy before. So that's what's nice about the auto injection. Auto injection is you have full control over it. So if you want to slow down or stop, you're able to because it's just your own foot pedal controlling it. Generally, just for numbers' sake, I like to set the injector to around 12 to 14 PSI. So that's the rate. It should be a very slow drip of the gene therapy into the subretinal space. So having that control and not depending on somebody else is very, very nice with the auto injector. The one drawback is that you do need special equipment to be able to use that. You need an adapter to be able to put the gene therapy into it and then attach it to the same part on your vitrectomy platform that you would normally hook up silicone oil. And that's how it works. Those are very readily available in these kits, so it's not that big of a challenge. And I think both of them work well. It's probably split pretty evenly across the community. Half of us probably inject manually and half of us probably inject through the auto inject. Another question from Taylor Bradshaw how has insurance coverage played a role? I think this is a really, really important question because when you see the price tag on the gene therapy that we have right now, $500,000 in the United States, anybody would be able to afford that. And yet we develop these treatments because we want to get them to patients who need them. So that's a really important question. We have had through Spark Therapeutics, they've had a very robust benefits investigation arrangement through the payers. So that was all arranged even at the time of approval before it really commercially launched. They had a lot of agreements already in place with the different payers. So from a provider standpoint, essentially all I need to do when I want to give Luxturna to a patient is I work with Spark Therapeutics to initiate the patient investigation process. And there are actually people who know a lot more about all the steps and the process that it entails to negotiate with the payer and just make sure that all of the ends are tied before we're able to deliver this. So it is an area that I think is going to need to be built on, especially if we do end up with more gene therapies down the line, because we need to figure out a way to make sure there's coverage for these patients and coordination for those processes, which can be pretty resource and time intensive. So thanks very much for that question. Let me take a look here let me take one more question. I'm just going to scan this list really quickly. Give me one moment. Thank you for all the questions. I'm sorry I'm not going to get to all of them, but like I said, I've left my email there and please feel free to I welcome any questions. Feel free to email me after our program today. I'm going to take a look at all right, I'm going to take one more about subretinal and suprachoroidal injections. And this person is asking which one will be the future trend, which one is better. Like I mentioned before, we don't have an answer to that question yet, unfortunately. So subretinal has been studied much more extensively than suprachoroidal injections, but both of them have their pros and cons. Subretinal injections seem, at least so far, to be the most effective when I'm looking across kind of broadly across all trials. I don't want to speak for any given product, but they're very effective. They're very safe in general and well tolerated because it is an immune privileged space in that subretinal space, but they are more challenging to give because you need a vitrectomy, you need to either tunnel through the suprachoroidal space or perform a vitrectomy, which really can be a disadvantage in younger patients. Suprachoroidal injections, I think, are really promising, and we already have an approved agent to give steroids, not gene therapy, but steroid treatment in the United States for uveitic macular edema, for example. So it is being done. It's nice because you can give it in the clinic and it's very straightforward for the most part. However, we're not yet sure we don't fully understand the efficacy profile of that in comparison to subretinal gene therapy approaches. So I think the ABVV RGX-314 studies are going to be really important because they're actually taking the same gene therapy and looking at it both ways. We're going to learn a tremendous amount both about efficacy as well as safety from that trial. So stay tuned. Like I said, I think all of these routes deserve continued treatment so that we can learn eventually what the optimal approach is. And like I said earlier, it may vary depending on the disease itself. You may want to give certain treatments subretinally and other treatments suprachoroidally. We're going to find out. Once again, thank you so much for all of the interest, for being on, spending part of your morning, afternoon, evening, or late evening with me here today. All of the engagement with the questions here. Like I said, feel free to reach out if you have any comments or additional lingering questions that we weren't able to get to. Thank you once again to CyberSight and Orbis International for the invitation to come today. Take care.